A warm welcome to this platform. So we continue with revision. As you know, we are going to revise or go through all these topics that you are able to see and also some more. So this is the second session that you are meeting, and of course our revision continues. So let us see what we can do in this one. All right, so for today we start on transport transport across the membrane, and here are the questions that we are going to answer. Okay, so transport across the membrane, what is it? So materials, let me just briefly explain a thing before we even go into the question, because it's very important. It will make every work that we are going to discuss very easy. I like it doing it that way. So when you talk of transport across the membrane, there are three processes by which the materials cross the membrane. One is called, so I can say, transport across the membrane. So there are three ways materials, or three processes in which material can cross the membrane. So one of them is osmosis. Osmosis is just the movement of water molecules from high water potential to the region of lower water potential, of course, across the permeable membrane, or it's the movement of water molecule from a dilute solution to the concentrated solution. The other one that we can talk of is diffusion, which is just the movement of particles from the region of higher concentration to a region of lower concentration. Then, of course, we can talk of active, that is active transport. Active transport has to do with the movement of materials against the concentration gradient. And the difference between active transport and the two, that is osmosis and diffusion, is that during active transport, or the process of active transport, requires energy. So that is one of the questions that you expect. Okay, that is one of the questions that is viable for the exams. Okay, the difference between, let's say, osmosis and diffusion. Okay, so this involves energy. Now, these processes, you need to know them very well. And as we go through the questions, we'll get to emphasize on this. But let us dwell on osmosis. Let us dwell on osmosis. So what causes the net movement of the water molecules from the cell to outside the cell, or from outside the cell to the inside cell? So now here we look at the effect of osmosis. Effect of osmosis on plants and animal cells. Now, to discuss this part, it's very important that we know three types of solutions in which the cell can find itself. That is plant or animal cell, in which you can put a cell. So types of solutions, we have what we call hypertonic solution. And I think this is something that you really need to understand here. A bit confusing, but something, if you get it, you have no problem. Then we have hypotonic solution. Then we have isotonic solutions. These you need to know how to define them. So hypertonic solution is the one that is more concentrated compared to the inside cell. So you always compare to the inside cell, which is the sap of a cell. So hypertonic solution is more concentrated, which means the inside cell is less concentrated and it has high water potential. There's a net movement of water from inside the cell to outside cell. And this has different effect on plants and animal cell, as we are going to see. Then hypotonic solution is the one that is less concentrated, or it has high water potential compared to the inside cell. Therefore, there's movement of water into the cell. Okay? Then isotonic solution, there's equal concentration. That inside of a sap, or that inside the cell, and also outside the cell. In other words, there's equal concentration. So sap, when I talk of sap, is just a component of the vacuole that is in the plant cell. But of course, in the animal cell, it's a different thing. But anyway, I think this is a brief introduction that you need to know. Of course, as we get going, I'll get to give some more details on this. So let us have a look at the first question. Figure 1.1 and 1.2 shows freshly cut potato chips suspended in solution E and F at the beginning of an experiment and after 2 hours respectively. Now, one thing that you should understand, first thing first, what type of a cell are we dealing with? So a potato chip is a plant cell, or it contains plant cells. So everything that we are going to discuss here, everything should be centered around what happened to the plant cell when it is put in different solutions, of which we don't know. We are yet to establish that. I guess you are able to see my diagrams. Explain the differences in appearance of the potato chips in solution E and F after 2 hours. So the setup here, we have the freshly cut potato chips. And now we have solution E and also solution P, or solution F. Okay, solution F and solution E. This is the beginning of experiment. Potato chips are of the same size, but put in different solutions. After 2 hours, this is what happened to the potato chip in solution E, and this is what happened to the potato chip in solution what? In solution F. The first question here is explain the differences in appearance of the potato chips in solution E and F after 2 hours. Explain the differences in appearance. In appearance, we are looking at the physical appearance, what you are able to see. I know you know that when the cell or when the cells, yes, lose water, they shrink. But here you are looking at the appearance, or rather they reduce in size. But here you are not going to use what you know, but the appearance that is here, lest you miss the point. So what do you write? So you can see the potato chip in solution E reduced in size. Okay? What is the reason? Could be due to loss of water by osmosis. So we can say the potato chip in solution E reduced in size due to loss of water by osmosis. Okay, whereas or while the potato chip, okay, whereas the potato chip in solution F increased in size due to osmotic gain of what? That or this is the difference in appearance. So for that you are given the marks. Now let us look at the second one, that is A2. Based on the appearance of the potato chips in solution E after 2 hours, what term could be used to describe solution E? So the solution E is the one. Now, which one of these? Is it hypertonic or hypotonic? Let us go back. The one that is the one that is more concentrated outside here and less compared to the inside cell is the kind of solution that we are dealing with here. What is the reason? Because there was a loss of water, or the cell lost what? So the kind of solution without wasting time here is called hyper. The term that is used to refer to the solution is called hypertonic solution. Okay? Then what would be, that is in B, what would be the state of the cells in potato chips in solution F after 2 hours? What would be the state of the cells? So pay particular attention the way you answer the questions. You might have answers, but of course you might miss a thing. So the state of a cell in potato chip, in the one that was put in solution F after 2 hours, you can see there's a swelling of the potato chip. So you expect the cells to be turgid. So there, the cells will be turgid. Okay, turgid. Then now let us go to the next question. State the importance of the state of cells in B above in plants or to plants. So this enables the young plants to stand firmly. Okay, enables young plants to stand firm. Okay. Then explain the meaning of the term isotonic solution. So from my earlier explanation, I've already explained. So isotonic solution is the one that has equal concentration to that of inside what? Inside the cell. So they have equal concentration to the inside cell. That is what you call isotonic solution. Let us have a look at the second question. So figure 1 shows the changes that take place. So that is taking place when the plant cell is placed in the solution of a known concentration from stage 1 up to stage 3. Identify the parts labeled A and B. So what are the parts labeled A and B? So the part labeled A is the cytoplasm, and the part labeled B is the vacuum. Using the changes in figure 1, explain what could be the concentration of a solution in which the cell was placed compared to the cell sap. So this is the cell was put in solution, and these were changes. Okay, you can see the detachment of the cell membrane from the wall or from the you can see the detachment of the cytoplasm. Okay, so here what is the concentration? So if there's detachment here, which means there was loss of water. So which means the inside cell was less concentrated compared to the solution. So we can say the cell sap was less concentrated compared to the solution. Let us read the question once more again. Using the changes in figure 1, explain what could have been the concentration of a solution which the cell was placed compared to the cell sap. So the concentration, you can also write it this way: the concentration of the solution was highly concentrated, or the solution was highly concentrated compared to the inside or the cell sap, or the cell sap was less concentrated compared to the solution. Then what term is given to the process that led to the changes in figure 1 from stage 1 to stage 3? Now look at this. Here they are saying what process. Okay, so this process is called cell this is osmosis. Osmosis is the process that resulted into that, because the cell lost water. What could be the overall effect of this process on the plant? It will result into wilting, okay, because the cells becomes flaccid, okay, which we call cell plasmolysis. So it will result into wilting, but the long effect, it will result into drying of a plant, okay. There's temporal wilting and permanent wilting. Then let us look at this question. Figures show red blood cells placed in different solutions. So here now we have a situation where the animal cell is placed in different solutions. So I have this solution, solution A and solution B. State the types of solution in which each cell was placed. State the type of solution in which each cell was placed. So look at this. This red blood cell, after put in solution A, it reduced in size. This is shrinking. So which means it lost water. Can only be possible if the solution is highly concentrated compared to the inside cell. So cell A was put in hypertonic solution. Then of course when it comes to cell B, was put in what? Hypotonic solution. The cell gained water and hence result into what you call cell lysis. Okay, that can only be when it gains a lot of water. Unlike the plant cell, which has a cell wall which is made of tough materials, that is cellulose, it cannot burst, but instead it just become turgid. But in the case of animal cell, they tend to rupture or undergo cell lysis when they gain a lot of water. And that's a part also you need to understand very well. Give an explanation for your answer. Okay, why are we saying the cell A was put in a hypertonic solution? So the reason behind that is, so the reason for the first one, so cell A was put in hypertonic solution because it lost water by osmosis, resulting into cell crenation. Okay, resulting to cell crenation or shrinking. Then cell B was put in hypotonic solution. What is the reason? Because it gained water by osmosis, resulting into cell lysis. Okay, that is it. Then, away from transport across the membrane, let us look at some enzymes. And now what do you need to know about enzymes? You need to know about characteristics of enzymes. So just to give you the guide on what you need to know, characteristics of enzymes. What are the characteristics of enzymes? One, enzymes are protein in nature. They catalyze forward and backward reaction. They speed up rate of chemical reaction. Also, they are specific in nature, which means they act on the specific substrate, and some more characteristics. So have a look at the characteristics of enzymes before you go in an exam. Aside from that, you also need to know about the enzyme specificity, that is lock and key, and also the structure of an enzyme. Be able to identify the active site and of course the allosteric site. What are the factors that affect enzyme activity? You talk of temperature, pH, enzyme concentration, substrate concentration, inhibitors, cofactors. Those you should know them. Then also industrial application of enzymes such as baking and brewing, tanning, talk of treatment of waste materials, quite a number of uses of enzymes. So you also need to know those industrial application of enzymes. Now, back to the question. Figure 2 shows the stages involved in an enzyme catalyzed reaction. And by the way, also before we go further, you need to know the categories of enzymes. We have intracellular and extracellular enzymes. Okay? Now, from stage 1 in figure 2, identify the parts enzyme. So figure 2 specifically, this, or stage in stage 1, in figure 2. So the whole of this is figure 2. Then stage 1, identify the enzyme. So the enzyme is letter I. Then the substrate is G and H. Which stage shows enzyme substrate complex? So enzyme substrate complex is where the enzyme, substrate are bound, or the substrate is into the active site. So it's stage 2. Label the active site on figure 2. So label the active site. So this is the active site where the substrate bind. This is the active site. Then label the active site, or just labeling the active site. Yes. Then see what type of metabolic reaction is catalyzed in the figure 2. So there are two types of metabolic reaction. We have what we call anabolic reaction, which involves the building of large molecules from smaller ones. Then we have what we call catabolic reaction. Catabolic reaction, for it has to do with breaking down of large molecules into smaller ones. So here you have this part and this part. Look at the end, they are attached, so that itself is building, and therefore this is called anabolic reaction. Okay, anabolic reaction. Then Name 2 regions in the human body where such metabolic reaction above occur. So where does such metabolic reaction occur? So 1, in the liver, there's a building of that is glucose to glycogen, that is a conversion. So one part, that is where they occur. But when you talk of breaking down, along the alimentary canal, the food nutrients are broken down into their simpler units. State the general term used to refer to enzymes that work inside the cell. So like I said, 2 types of enzymes. We have intracellular and extracellular. The intracellular enzyme is the one that works from where it is produced. It works from within the region where it is produced, in other words. That is what we call intracellular. But extracellular, they are produced, and of course, they have a region now where they are moved to perform the function. So the one that works inside cell is called intracellular enzymes, intracellular enzymes. Then describe the 2 industrial application of enzyme. Like I said, industrial application of enzyme, you need to know them. So you can talk of baking and brewing. What is the other one that I gave? So be able to put it here. All right, let us look at the second question. Figure 2 shows the effect of pH on rate of enzyme catalyzed reaction. What term is given to J and L? The figure 2.1 shows the effect of pH. So we are dealing with pH. So like I said, the factors that affect enzyme activity, one of them is pH. So what term is given to J and L? So J and L, this is the top part of the graph. So those are called optimum pH. Optimum pH. Explain why the rate of enzyme or rate of reaction K and M goes down after J and L. So this is the optimum pH. Now, you know what the optimum pH is? The optimum pH is as a temperature. I mean, that's a pH at which the enzyme has maximum activity. Okay, it works well at that point. Beyond that, it is beyond its optimum pH. And of course, the enzyme starts denaturing. Okay, so why the rate of reaction reduces? Because the enzyme start denaturing. Okay, start denaturing, or the enzyme is denatured, so it cannot perform its function fully. Name the region of the alimentary canal where reaction K and M take place. So region, if you look at the pH for this one, is around 2 point something here, because it's between 2 and 3, so it can say 2.5. Now, in terms of pH, this is an acidic pH. You know the pH scale runs from here 0, 14. So this is 7. Less than 7, pH is acidic. Above, that is alkaline. So the region of the alimentary canal for K is the stomach, because that is the acidic, or that is where you find the reaction that take place at acidic pH. But of course, M, we can talk of the what? The duodenum. At that particular point, or in that region, the reactions occur at alkaline pH. Okay, so we have K, the regions, stomach, and duodenum. Okay, then suggest one enzyme that can catalyze reaction M. So in the duodenum, you find enzymes such as peptidase. Okay, you find enzymes such as sucrase, maltase. So any of those. State one food nutrient which can be catalyzed by K. So in the stomach, digestion of proteins occur. So already here we can talk of proteins. Okay, pepsin work well at that temperature, at that pH. State two factors other than pH which affect enzyme activity. So this we said temperature, and also we can talk of enzyme concentration. Enzyme concentration. Okay, I guess you've picked that. All right, let us have a look at this one. So also it's a graph, but of course this time around is on temperature. So figure 2.0 shows a graph showing the effect of temperature on enzyme catalyzed reaction. What is an enzyme? So what is an enzyme? So an enzyme is just a biological catalyst that speed up rate of chemical reaction in living organisms or in the body. That is what an enzyme is. Okay, so that is an enzyme, and I hope no one really have difficulties on defining the enzyme. Then what is the optimum temperature of the enzyme reaction shown in the graph? So this is the plot of temperature against rate of reaction. Now, as the temperature increases, also rate of enzyme reaction increases until at a particular temperature, otherwise known as optimum temperature, and is found on top there. So at that particular point, the enzyme has maximum activity, but beyond that, the temperature, when the temperature increases beyond that, the enzyme starts denaturing. So at what temperature is the enzyme completely, or what temperature, what is the optimum temperature? So you look at this part here. So that part I can approximate it to be 30. If that is 40, so I can approximate it maybe 36, 35.5. I think it should be accepted. But I'm not saying that I'll write all these. I'll just write 36 degrees Celsius. Then at what temperature is the enzyme completely denatured? Now, denaturation starts from here. So the enzyme start denaturing until at a point where it touches the temperature part. At that point, the enzyme has zero activity, or the rate of enzyme activity is 0. Okay, which means the enzyme is completely denatured. Now they're saying at what temperature? So this is 55. Up to this, I can approximate it to be 50. Let me say 50. Not 56. Let's say 57.5 degrees Celsius. But of course, that is an approximate. Then what is the reason? The reason I've already given there, because at that particular point there is zero rate of reaction, okay, or the enzyme has 0 activities. That is the reason why we're saying that part the enzyme is completely denatured. Explain why it is important to measure the body temperature of the person when the person is sick. Okay, explain why. Now you should know that different conditions or different sicknesses show different body temperature changes. So one of the reasons to determine the body temperature, explain why it is important to measure the body temperature when the person is sick. So one of the reasons we can say to determine the medication to give the person. Okay, so to determine the medication to administer to the patient. What could be the other one? Okay, be able to suggest here. Propose 2 ways the body temperature of a sick person can be brought to normal. Okay, so this question is kind of general. So what are the ways? What do you think? What are the ways? So also this one, I leave the question to you so that even as you go through, you'll be able to do a bit of research as well. All right, I hope up to this point it has been beneficial. So the next one, we are going to look at, we are going to start looking at digestion. And of course, we'll cover also a topic ahead of this. Otherwise, thank you for staying up to this point. But please, if you are interested to join us for physical lessons, we are found in Kawata, just near Kawata Primary School. And also, if you want to join for online lessons, you can still get in touch on this number. Otherwise, see you in the next one.